Before starting this lecture, I just want to give you one information that the images, text and other materials used in our presentation are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the same. So in this lecture, we'll talk about sex determination. Then we'll talk about mutation. And we'll talk about the genetic disorders also. And we'll discuss the practice questions related to these topics. So let's begin with this lecture. So we'll discuss sex determination first. The mechanism of sex determination has always been puzzled before the geneticist. The initial clue about genetic or chromosomal mechanism of sex determination can be traced back to some of the experiment carried out in the insects. So initial experiments were carried out on insects, and these experiments were related to the genetic or chromosomal mechanism of sex determination. Means the sex, is it determined by the chromosomes or the genes which are present on the chromosomes? So is there any genetic or chromosomal basis of sex determination? Now, in fact, the cytological observation made in a number of insects led to the development of the concept of genetic or chromosomal basis of sex determination. Now, the cytological observation was made in the insects. So they looked at the components of the cell. The cell has a nucleus, and within the nucleus, these chromosomes are present. So by cytological observation done in the insects, this led to the development of the concept of genetic or chromosomal basis of sex determination. Now, Henkin in 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects. So what is spermatogenesis? Formation of sperms. And it was also observed by him that 50% of the sperm receives structure after spermatogenesis, whereas the other 50% sperm did not receive it. So there was one structure that was present in 50% of the sperm, and the structure was absent in other 50% of the sperm. Henkin gave a name to this structure as the X-body, but he could not explain the significance of this X-body. Why X-body is present? What is the purpose of this X-body? Now, further investigation by other scientists led to the conclusion that the X-body of Henkin was in fact a chromosome, and that is why it was given the name X chromosome. So later on, it was found that this X body was a chromosome, and this was named as X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of the XO type. That is, all eggs bear an additional X chromosome beside the other chromosomes, which are the autosomes. On the other hand, some of the sperm bear the X chromosome, whereas some do not. So in the insects, some of the sperms, they bear X chromosome, whereas some of the sperm lacked the X chromosome. That's why the system is X0 type or XO type. This O or 0 represents the lack of the X chromosome. So in insects, the female, the eggs always bear an X chromosome. Okay, and there is additional copy of an X chromosome beside the other autosomes. But in the sperms, in insects, half of the sperm will bear X chromosome and half of the sperm don't bear the X chromosome. Now let's discuss this question. Which scientist first identified the structure later known as X chromosome? What are the options? Option number A is Morgan, B is Henkin, C is Mendel, and D is Watson. Answer is option number B, Henkin. Let's have a look at the explanation. Henkin was the scientist who first identified the structure and named it the X body, which was later understood to be the X chromosome. Let's have a look at the explanation of the incorrect option. Option number A, Thomas Hunt Morgan is known for his work on fruit flies and the discovery of sex-linked inheritance, but he did not identify the X body. Option number C, Gregor Mendel is known as the father of the genetics for his work on pea plant, but he did not identify the X body. Now option number D is incorrect. James Watson is known for the discovery of the DNA double helix structure, not for identifying the X body. Now, eggs fertilized by sperms having an X chromosome become females, and those fertilized by sperms that do not have an X chromosome become males. Do you think the number of chromosomes in the male and the female are equal? So in insects having the X O chromosome, the number of chromosome will be more in the females as compared to males. And due to the involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome. And the rest of the chromosome were named as the autosomes. So X chromosome was called the sex chromosome because it was involved in sex determination. So grasshopper is an example of X0 type of sex determination in which the males have only X chromosome besides the autosomes, whereas the females have a pair of the X chromosome. So in females, a pair of X chromosome is present. In males, only one X chromosome is present besides the autosomes. These observations led to the investigation of a number of species to understand the mechanism of the sex determination. In a number of other insects and mammals, including man, XY type of sex determination is seen, where both male and females have same number of the chromosome. Like in humans, both females and males, they have 46 chromosomes. Among the males, an X chromosome is present, but its counterpart is distinctly smaller and is called the Y chromosome. So Y chromosome is smaller, it is shorter than the X chromosome. Females, however, have a pair of X chromosomes. Both male and female bear same number of autosomes. So in humans, in man, total are 46 chromosomes, means 23 pairs. Out of 23 pairs, 22 pairs are that of the autosomes. Now, hence, the males have autosomes plus XY, while females have autosome plus XX. So in total, in humans, we have 46 chromosomes. Let's discuss this question. Which of the following statement is true regarding the sex determination system in humans and many other mammals? So what are the options? Option number A, males have 2 X chromosome. Option number B, females have 1 X and Y chromosome, 1 Y chromosome. Option number C, males have 1 X and 1 Y chromosome. And option number B, both males and females have the same sex chromosome. Answer is option number C. Males have one X and one Y chromosome. Let's have a look at the explanation. Males have one X and one Y chromosome, and this is the basis for the XY type of sex determination in humans and many other mammals. Let's have a look at the explanation of the incorrect option. Option number A is incorrect. Males have two X chromosome. This is incorrect because males have one X and one Y chromosome, not two X chromosomes. Then option number B is incorrect. Females have one X and one Y chromosome. This is incorrect because females have two X chromosome, not one X and one Y chromosome. Then option number D, both male and females have the same sex chromosome. This is incorrect statement because males and females do not have the same sex chromosome. Males have XY and females have XX. Now in human beings and in Drosophila, the males have one X and one Y chromosome, whereas females have a pair of X chromosome beside the autosomes. Now, in the above description, you have studied about two types of sex determination mechanism, that is X0 type and XY type. But in both the cases, male produce two different type of gametes. So, male are producing two different kinds of gametes here in both these systems. So, now in the first one, either with or without the X chromosome, like in the X0 type, and some gametes with X chromosome and some with the Y chromosome in the XY type of sex determination. Such type of sex determination mechanism is designated to be the example of male heterogamety. So this means, what is male heterogamety? Means males are producing two kinds of gametes here and females are producing same kinds of gametes here. So this is showing both of these systems, they are showing male heterogamety, means males are producing two kinds of gametes, and they are also showing female homogamety, means females are producing only one type of gamete. Now, in some other organism, for example, bird, a different mechanism of sex determination is observed. So you can see in this figure, okay, this figure A, this is showing you the chromosome in the humans. So you can see here, this is the determination of sex by chromosomal inheritance. A and B, both in the humans and in the Drosophila, the female has a pair of XX chromosome. Means they are homogametic, they are producing only one kind of gametes. All the gametes will have the X chromosome. And the male are XY, means they are heterochromatic, they will produce two kinds of gamete. Half of the gamete will contain X chromosome, half of the gametes will contain the Y chromosome. In many birds, okay, female has a pair of dissimilar chromosomes ZW and the males have two similar ZZ chromosome. So you can see this is X chromosome, Y chromosome is little shorter, females have a pair of X chromosome. Similarly in Drosophila also, males have X chromosome and the Y chromosome is little bit shorter and the females have a pair of X chromosome. In Drosophila you can see the male is shorter in size, smaller in size compared to female. In birds, the sex determination system is little bit different. Here you can see the females, you can see they have the Z and W. W is little bit shorter than the Z chromosome. Okay, so female is the heterogametic sex here and males, they are homogametic. Males are producing one kinds of gamete here. All the gamete will have the Z chromosome. In females, you can see two kinds of gametes are produced. One will contain, half of the gametes will contain Z and half of the gametes will contain the W chromosome. Now let's discuss this question. In human beings, what determines the sex of an individual? So what are the options? Option B is number of autosomes, B is presence of the Y chromosome, C the number of the X chromosome, and D the presence of two X chromosome. Answer is option number B, the presence of the Y chromosome. Let's have a look at the explanation. In humans, the presence of the Y chromosome determines maleness. The males have one X and one Y chromosome, means XY, while the females have two X chromosome, means XX. Let's have a look at the explanation of the incorrect option. Option number A, autosomes do not determine sex. They are the non-sex chromosome. Option number C is incorrect. Number of X chromosome is important. It is the presence of Y chromosome that determines the maleness. Option number D is incorrect. Presence of two X chromosome determines femaleness, not the maleness. Now, in this case, the total number of chromosome is same, but in both males and females. But two different type of gametes in terms of sex chromosomes are produced by females, that is female heterogamety is seen here. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosome of the female bird has been designated to be Z and W chromosome instead of X and Y. In these organisms, the females have one Z and one W chromosome, whereas males have a pair of Z chromosome beside the autosomes. Now let's discuss the sex determination in humans. So it has already been mentioned that the sex determining mechanism in case of humans is XY type. Out of 23 pair of chromosomes present, 22 pairs are exactly same in both males and females, and these are the autosomes. A pair of X chromosomes are present in the female, whereas the presence of an X and Y chromosome are determinant of the male characteristics. And during spermatogenesis, means sperm formation among males, two types of gametes are produced. 50% of the total sperm produced carry X chromosome, and the rest 50% will carry the Y chromosome beside the autosomes. Now let's discuss this question. How many pairs of autosomes are present in humans? What are the options? Option number A is 22, B is 23, C is 1 and D is 2. Answer is option number A, 22. Let's have a look at the explanation. So humans have 23 pair of chromosomes in total, out of which 22 pairs are autosomes and one pair is the sex chromosome. Means, let's have a look at the explanation of the incorrect option. Option number B, 23 includes both autosomes and sex chromosome. Option number C, 1 is incorrect as there are 22 pair of autosomes. Option number D, 2 is incorrect as there are 22 pair of autosomes. Now females, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. Now, in case the ovum or the egg fertilizes a sperm carrying X chromosome, the zygote develops into the female, means it will have XX chromosome, a pair of X chromosome. So, what is fertilization? Fusion of the male and the female gamete. So, after fertilization, zygote is formed that will develop into the baby. So, if the ovum fertilizes with the sperm carrying X chromosome, the zygote will develop into female, and the fertilization of ovum with the Y chromosome carrying sperm results in a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child, not that of the egg. It is also evident that each pregnancy, there is always 50% probability of either a male or a female child. It is unfortunate that in our society, women are blamed for giving birth to female children and have been ostracized and ill-treated because of the false notion. But it is male who determine the sex of the child. Let's discuss this question. What determines the sex of the child? What are the options? Okay, if we talk about humans, who determine the sex of the child? What are the options? Option number A is genetic makeup of the ovum, B genetic makeup of the sperm, C is the environment, and D is the mother's diet. Answer is option number B, the genetic makeup of the sperm. Let's have a look at the explanation. Now, the sperm can carry either an X or a Y chromosome, which determines whether the zygote will be female, means having a pair of X chromosome, or a male having X and Y chromosome. Let's have a look at the explanation of the incorrect option. Option number A is incorrect. Ovum always carries an X chromosome, so it does not determine the sex of the child. Option number C, the environment does not influence the genetic determination of the child's sex. Option number D is incorrect. Mother's diet does not affect the genetic determination of the child's sex. Now let's discuss the sex determination in honeybee. Sex determination in honeybee is based on the number of set of chromosomes an individual receives. An offspring formed from the union of sperm and egg develops as a female. So females develop after the fertilization, and the female can be queen or the worker bee. And an unfertilized egg develops into male. So males are produced without fertilization, and they are the drones. And development of an organism without fertilization, this is known as parthenogenesis. The males develop due to parthenogenesis. Okay, so this means that the males have half the number of chromosomes than that of the females because they are produced parthenogenetically. Now the females are diploid, having 32 chromosomes, and the males are haploid. Okay, that is, they will be having 16 chromosomes. So this is known as haplodiploid sex determination system and has special characteristic feature such as the males produce sperms by mitosis. So usually these sperms are produced by meiosis, but here males are haploid, so they produce sperm by mitosis. Now they do not have father because again they are produced by parthenogenesis. They are developed from unfertilized egg, and thus they cannot have sons also. But they can have a grandfather and can have grandsons. So here let's discuss this image 4.13. This is sex determination in honeybee. These are the parents, female and the male. Parents here you can see females have 32 chromosomes, males have 16 chromosomes. Males are haploid, females are diploid. Females through meiosis produce the eggs. Males through mitosis will produce the sperms. The sperms will contain 16 chromosomes. And the gametes, female gametes, eggs will also contain 16 chromosomes because they are produced by meiosis. Now, males, they develop by parthenogenesis. So, males are haploid here. Females, they are developed after fertilization. So, the egg containing the 16 chromosome will fertilize with the sperm containing 16 chromosomes. So, females will have 32 chromosomes. They will be diploid. So, this is the haplodiploid sex determination system in the honeybees. Now let's discuss this question. In the haplodiploid sex determination system of honeybees, how many chromosomes does a male drone have? What are the options? Option number A is 16, B is 32, C is 48, and D is 64. Answer is option number A, 16. Let's have a look at the explanation. In the haplodiploid sex determination system, males, which are the drones, are haploid and have half the number of chromosomes compared to females. Since females are diploid with 32 chromosomes, males have 16 chromosomes. Let's have a look at the explanation of the incorrect option. Option number B. So 32 is incorrect because 32 chromosomes is the diploid number found in the female honeybees, not males. Option number C is 42 is incorrect because 48 chromosome is not a characteristic chromosome number in honeybees. Option number D, 64 is incorrect because 64 chromosome is not a characteristic chromosome number in the honeybees. Now, how is sex determination mechanism different in the birds? Now, is the sperm or the egg responsible for the sex in the chicks? A mutation. Let's discuss mutation. Mutation is a phenomena which result in the alteration of the DNA sequences. Means there is a change in the DNA sequences, and consequently, this result in changes in the genotype and the phenotype of the organism. So, a gene is responsible for a particular trait, for a particular phenotype. So, if the sequence of the DNA sequence is changed, then what happen? The protein which it codes for, it will change, because the codons, they code for a particular amino acid. So if the sequence of the DNA changes, then what will happen? It might code for some other protein which can give some other phenotype. So due to mutation, the genotype and phenotype both can change. In addition to recombination, mutation is another phenomena that leads to variation in the DNA. So variation can arise due to recombination, which happens during meiosis, and it can arise due to mutation also. Now let's discuss this question. Which of the following is responsible for determining the sex of the chicks in the bird? What are the options? Option number A is sperm, B is egg, C is both sperm and egg, and D is neither sperm nor egg. Answer is option number B, egg. Let's have a look at the explanation. In bird, the sex of the offspring is determined by the egg. The female bird have ZW sex chromosome, while the males have ZZ sex chromosome. The egg from the female bird can carry either a Z or a W chromosome, while the sperm from the male bird always carry a Z chromosome. Therefore, the egg determines the sex of the chick. Let's have a look at the explanation of the incorrect options. Option number A is incorrect. Sperm is incorrect because the sperm from the male bird always carry a Z chromosome and does not determine the sex of the chick. Option number C, both sperm and eggs, is incorrect because only the egg, which can carry either a Z or W chromosome, determines the sex of the chick. Option number D, neither sperm nor egg, is incorrect because the egg is responsible for determining the sex of the chick. Now let's discuss about mutation. Now you must have learned that one DNA helix runs continuously from one end to the other in each chromatid in a highly supercoiled form. Therefore, loss means deletion, or gain means insertion or duplication of a segment of the DNA, result in alteration in the chromosome. So since genes are known to be located on the chromosomes, alteration in chromosome can result in abnormalities or aberrations. Now, chromosomal aberrations are commonly observed in the cancer cells. In addition to the above, mutation also arise due to change in the single base pair of DNA known as the point mutation. So, a classical example of such a mutation is sickle cell anemia. We'll talk about sickle cell anemia in detail in upcoming slides. Now, deletion and insertion of base pair of DNA causes frame shift mutations. The mechanism of mutation is beyond the scope of discussion at this level. However, there are many chemical and physical factors that induce mutations. So these are referred to as mutagens. So what are mutagens that induce mutations? Now UV radiations can cause mutations in organism. So it is a mutagen. So UV radiation, they are physical mutagen. So mutagens, they are of two types, chemical mutagen and physical mutagen. Okay, so these chemical mutagens, they are known as the alkylating agents. So some of the alkylic agents, they act as chemical mutagens and they alter the DNA sequence of the chromosomes. Now let's discuss this question. Which of the following is an example of point mutation? What are the option? Option number A, deletion of a base pair, B is insertion of a base pair, C is sickle cell anemia, and D is UV radiation. Answer is option number C, sickle cell anemia. Let's have a look at the explanation. Sickle cell anemia is a classical example of point mutation which arises due to change in a single base pair of DNA. Now in this case, mutation occurs in the hemoglobin gene, leading to the production of abnormal hemoglobin proteins. Let's have a look at the explanation of the incorrect option. Option number A is incorrect. Deletion of a base pair is an example of frame shift mutation, not a point mutation. Option number B is incorrect. Insertion of a base pair is also an example of frame shift mutation, not of point mutation. Option number D is incorrect. UV radiation is a mutagen that causes mutations but not an example of a point mutation itself. Now let's talk about the genetic disorders, and within this we will talk about the pedigree analysis. Now the idea that disorders are inherited has been prevailing in the human society since long. Now this was based on the heritability of certain characteristic features in the family. Certain characteristics, they are inherited from the parents to the offerings, and characteristics, they are controlled by the genes. After the rediscovery of Mendel's work, the practice of analyzing inheritance patterns of traits in human being began. So Mendel, he worked on the pea plant and he gave the laws of inheritance. Now the scientists, they wanted to study what is the inheritance pattern in the human beings, how the traits are inherited in human beings over successive generations. Since it is evident that control crosses that can be performed in a pea plant on some other organisms are not possible in case of human beings, the study of family history about inheritance of a particular trait provides an alternative. So we cannot perform control crosses in humans. It is not ethical also. Now, such an analysis of traits in several of generation of a family is called the pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. Let's discuss this question. In pedigree analysis, what is the primary purpose of studying the family history of a particular trait? What are the option? Option number A is to determine the exact genetic sequence responsible for the trait. Option number B, to analyze the inheritance pattern of trait across several generations. Option number C, to identify environmental factors affecting the trait. Option number D, to perform controlled genetic crosses similar to those done in pea plants. Answer is option number B, to analyze the inheritance pattern of traits across several generations. Let's have a look at the explanation. The primary purpose of pedigree analysis is to study the inheritance pattern of particular trait across several generations in the family. So this helps in understanding how the trait is passed down and whether it follows Mendelian inheritance patterns. Let's have a look at the explanation of the incorrect option. Option number A is incorrect. Pedigree analysis does not provide information about the exact genetic sequence. It focuses on the inheritance pattern. Option number C is incorrect. Pedigree analysis is concerned with genetic inheritance, not environmental influences. Option number D is incorrect. Controlled genetic crosses are not possible in humans. Pedigree analysis is an alternative method to study the inheritance patterns.